Okay, here is the first of, I'm sure, two lectures on neural tissue, chapter 12. Now, it's really important that you realize that we cover this in 231, because when you step into 232, you will be expected to know this, because you'll start right into the study of the brain itself. And if you don't know the tissues that make up the brain, and you don't know how the spinal cord connects to everything, because the spinal cord is kind of in between the body and the brain, then a lot of that won't make sense to you. So you got to know this, and I would recommend that you review it prior to 232, whenever you take 232, assuming you take it. Because you'll need it. You'll need it right there, sort of at the tip of your skull, or your tip of your tongue. I went out to Oaks Park one time a few years ago, and I watched dog jumping. Y'all ever seen this? This is the craziest thing. It's so cute. These dogs are having the greatest time of their life. And what they do is they have a little short running ramp they get to run on, and then they jump as far out into the water as they can, and they film them and they measure them to see exactly how many feet they got. And it just cracks me up that the owner sits up there and he throws something out to them that they kind of jump for. Most of them never catch it, but they can catch it after it hits the water. It's just amazing. This guy here, and you can see that the measurements over here along the wall here. And here's one getting ready to take off. He's just loving it. And there he goes. And then I love this picture here of one as he lands. So his neural cells are getting stimulated there, and that's a good thing for all of us from time to time. Basically, you need to know that neural tissue, when we talk about neural tissue, we're mainly talking about the tissue of the brain and the spinal cord. Obviously, there are some nerves that participate in this as well. But the thing to know is that neural tissue consists of two kinds of cells. It consists of nerve cells, which we call neurons, and they are the ones that carry electric sparks. They carry the action potentials, and they perform the work that the brain does, which is carrying messages. And then there's the other group of cells, which are called the neuroglial or the glial cells, and they are simply cells that support the brain cells. So it takes two kinds. It takes the brain cells and then all their supporting cast. And there's more supporting cast than there are brain cells, although I think in overall volume, I think they work out to be about half and half. Anyway, so what are the organs of the nervous system? We've touched on them a little bit to this point. The neural system will involve the first 1/3, actually more than that. The first half of 232 will all be about the nervous system. It'll be about the brain, and then it'll be about the organs of special sense, like the ears and the eyes and the nose and taste and so forth. Anyway, the organs of the nervous system consist basically of the brain and the spinal cord. Those two are called the central nervous system. The CNS is the brain and the spinal cord. Then there's all these things on the outside that are out there to pick up messages to send back into us, into the brain. And those are things like the sensory receptors of the special things like the eyes and the ears, taste, our orientation, semicircular canals, and all that jazz. And then there's all kinds of things that are out there right there in the skin, which measure touch and temperature and feel and all that. So these nerves are an organ of the special nervous system, and nerves are nothing more than bundles of axons. Write that down. Nerves are nothing but bundles of axons. Now they might have some satellite cells along with them, some helper cells, but basically that's what a nerve is. So we can divide the nervous system into the central nervous system, which, as I already told you, is the brain and the spinal cord. and then the peripheral nervous system, the PNS, which is everything else. I think I just said that. What are the functions of the brain, of the central nervous system? Well, it's to coordinate all of this data, okay? Now, what does that mean? Well, we get all this sensory data sent into us, like feeling and sound and vision and all that stuff. And all that stuff is sent up to our brain for our brain to think about it and decide what's important and what's not, okay? Our brain then decides if we want to do anything about it, if we want to move. In other words, do we want to move our eyeball muscles around and move our eyes? Do we want to walk across the front of the room? Do we want to reach out and touch something? Do I want to push on this button here? Those are all motor commands that your brain makes up and decides, oh yeah, we want to do that, and it does it. What it does is it just sends messages out to things like skeletal muscles here, and it makes the muscles contract in order for me to push the button. Now, what about these higher functions? Because we also do this thing called thinking. What does that mean? And I think it's the hardest concept to ever understand. I think it's the one thing that human beings have not really been able to figure out, is how we actually think. So let me try to explain intelligence, memory, learning, emotion, and so forth in a simple example. Many of you have probably had babies around the house, and you've watched babies when they're really small, especially before the age of about 3 months, and even up to the age of a year. And they're laying there in bed, and they're looking around, and they see Mama, and they listen to Mama, and they hear Daddy, and they talk to Daddy, and they're making cuckoo noises, and and they move around, and they kind of grab at things, and if they have some toys in their bed, they pick them up, and what do they do with them? They put them in their mouth. That's right. Why do they put them in their mouth? Well, as you're going to learn in this chapter, in the next chapter, the reason they do that is because we have more sensory fibers around our mouth than anywhere else. We have a lot in our fingers, but we have a lot in our mouth. So we can, when we put things in our mouth, even our fingers, we're getting a lot of input up to our brain. We have a lot of sensation fibers. And how do we learn? Well, how does this baby learn? Well, the baby sits there and he puts things in his mouth all the time, and he feels them when they touch his mouth, and then every time he touches one of those sensors, they send the message up and say, hey, something is touching your lips. But the baby doesn't realize that that's his lips at first, okay? Now, all of you know that if you reach up and touch your lips, you know that you just touched your lips. Now, how do you know that? Well, you know that because you practiced while you were laying there in the baby crib when you were babies, and you put all kinds of crap in your mouth and your fingers in your mouth, and it finally dawned on you that, hey, I'm touching my lip. Oh, that's a lip. And then you grow up a little bit, and you can actually look in a mirror, and you touch yourself in the mirror, and you think, oh my goodness, that sensation that's going to my brain actually came from my lip. Because look, that's what my lip looks like. Because now you can see it as well as feel it. You look at your mama about 100,000 times, and eventually your eyes, when you're first born, your eyes can't even focus. So babies aren't really seeing as much as mama and daddies like to think they're seeing, but they can see some. And as they get to be 6 and 7 months old, they can actually focus, and so then they really can see. And then they might say, Dada, or Mama, and you're so excited. Because finally, this kid has made a relationship between what he's seeing and this face, right? Your face. Why? Because he's seen it 100,000 times. And it kind of embeds in his brain and establishes a pattern, and he knows whenever he sees that pattern, oh, that's Mama. It may be almost mealtime. Oh, it may be almost time to get my diaper changed. Oh,
that's a good thing. And the baby starts to coo and smile, and that makes mom and daddy feel really good, and so forth. So these patterns are being established in your brain, these patterns of shocks, and they go circling round and round in your brain every time you see that face. Now, I'll bet every one of you right now could close your eyes and think about it and see your mom. Right? Every one of you could close your eyes. I can do it too. Because you close your eyes and you can, if you want to, you think about it and you think, oh, yeah, that's Mama. I recognize what she looks like. Why? Because you looked at her so many times your whole life, right? Assuming your mom is still alive. Mine died 5 years ago now. I miss her a lot. Anyway, so this is what this business of patterns is that are forming in your brain. And that's really all your brain does. Your brain sends these electric shocks around from a set of nerve cells, and it's done that so many times that you establish that pattern as something you know, as a memory, okay? And that allows you to sit and think about things. So you've studied your A&P book, and you looked at those pictures of those muscles enough times that you finally kind of embed that in your brain, and you establish a pattern, and then you know what that is. Does that make sense? Because I think that's one of the hardest things is to understand how do these silly nerve cells, all they are is a dendrite, bunch of dendrites, and an axon that stimulates another nerve cell, another nerve cell, another nerve cell, hundreds of thousands of them. In fact, some brain cells will have 20,000 other cells attaching to them. That's a lot of dendrites. But that's how many of them, a lot of them have, in order to establish these patterns that allow us to have things like intelligence, memory, learning, and emotion. The peripheral nervous system, all it does is connect these receptors on the outside to our brain. That's all they do. They either deliver sensory information in, or they carry motor commands out. Now those motor commands may be, okay, bend your arm. Those motor commands may be, okay, sweat. In other words, if your body's too hot, you may be sending messages out to your sweat glands and telling your sweat glands to sweat. Okay? Part of the way we maintain hemostasis, body temperature, and so forth. We have a very limited number of nerves that go out. In fact, you can write this down because this is something you ought to know. We have 31 spinal nerves. We actually have 31 pairs of spinal nerves. We have one on each side. One for every level of the vertebrae. So we have C1, C2, cervical 1, cervical 2, all the way down to your coccyx. We have 31 pairs. We also have 12 special nerves. They're not really special. The only thing different is that they come right out of the brain. They don't come out of the spinal cord. They come right out of the brain. That's the only difference in them. We're going to learn all 12 of the cranial nerves. And you're going to know what every one of them does and where every one of them goes. The cranial nerves are very important. But that's all the nerves we have. 12 pairs of cranial nerves, 31 pairs of spinal nerves, makes it pretty simple. Every nerve carries both afferent and efferent information. In other words, every nerve carries information out, efferent, exiting. That's the way I always remembered it. Exiting is efferent, E. That's to carry muscle commands. That's to carry gland commands like sweating and so forth out. Every nerve has an efferent. That was the efferent division. And the afferent division is approaching. Approaching, bringing information in. So these all carry sensory information. These carry motor commands. And there's my hint at the bottom. Afferent approaches, efferent exits. We're going to learn about the receptors, the different types of receptors that we have in our body that detect changes or respond to stimuli. A lot of these are neurons, a lot of them are specialized cells, a lot of them are very complex organs like our eyes and ears. Now, you're going to hear me tell a lot of stories as I go through this whole business about the tiger in the room. Because I can promise you right now, in fact, I'll let you start thinking about it right now. What would happen if a tiger walked through that door right now? A great big tiger. Now, you guys would all recognize it as a tiger, I suspect, because you've seen pictures or maybe you've been to the zoo. I'd like to think at your age, everybody knows what a tiger is, right? But just think, if you were a baby, you wouldn't know what a tiger was, and it probably wouldn't upset you at all. But because we think we're smart, because we know what a tiger is, and we've heard these terrible stories about how tigers eat people, we would probably all be somewhat panicked. And it would cause all these changes in our bodies, believe me. Everybody's heart rate would go up, everybody's pupils would dilate. You're like, holy smokes, there's a tiger in the room. We would start breathing faster. We would start releasing sugar into our system so that we can juice up our muscles so that we can get ready to run, or we can get ready to fight the tiger. We call that fight or flight, okay? But don't think every one of you is not going to react to a tiger walking in the room, okay? So that's what all this nervous system stuff. Now, why do we have those reactions? Well, because back before we had Highway 26 and before we had cars and before we had lovely buildings like this, we probably needed to know what happens when a tiger or a grizzly bear or something like that walks in the room with us, right? And so it was a matter of survival to be able to make all those organs work so that you could either get away from him or kill him and survive and carry the species on so that we could all be here today, thanks to our ancestors. All right, so we have receptors. Receptors are things that bring, sense the things on the outside and send the messages in, and then we have effectors which make things happen, muscles and glands and so forth. We also have another way to split up the nervous system is between the in the efferent division. We're dividing the system into the efferent as opposed to the afferent at this point. We have the somatic nervous system. Somatic comes from somites, which comes from muscles that contract, and we have both voluntary and involuntary muscles that contract. In other words, when that tiger walked in there, I immediately started doing this. And that's signals my brain is sending to my muscles to say, let's get the hell out of here. And we also have the autonomic nervous system. The autonomic nervous system is this thing that you can almost think of the autonomic system as the automatic nervous system. For example, when's the last time, Tamara, that you thought about your heart rate? You thought, oh my goodness, I'm feeling a little slowed down. I need to speed up my heart. When you went to the gym? So you think your brain thought about and said, oh, I'm going to exercise, so I need my heart to beat faster. The point is, you didn't think about it, did you? When's the last time you thought that, I had that breakfast sandwich this morning, and I need for my stomach to contract 3 or 4 more times to get it mooshed up so I can digest it, right? You didn't think about that. We don't think about stuff that our body just controls for us on its own. Not one of you has thought about your blood pressure. You go from sitting to standing, and you really do need your blood pressure to come up, because when you're standing, you've got to pump blood uphill all the way to your brain, whereas if you're sitting down or laying down, it's pretty easy to get blood to your brain, right? So our automatic nervous system, ANS, controls all of these subconscious things. We have a whole chapter on the autonomic nervous system in about 3 chapters, in 232. Okay. And we divide the autonomic nervous system into 2 parts, just so you know. You probably ought to know this at this point. One is called the sympathetic division. Now, this doesn't mean it has anything to do with sympathy. It just called that for some reason. I don't really understand why. It seems silly to me. But just know that the sympathetic nervous system is the stimulating nervous system, and it's the one that we would call the fight or flight, get cranked up, let's get ready to go type nervous system. And just on the other side of that, doing the opposite, we have the parasympathetic nervous system, and it has a relaxing effect. It's also called the rest and digest system. Now, by that I mean, when you're out running a marathon, anybody ever done that? Been on a long run? When you're on a long run, you don't send any blood to your stomach or to your intestines. You want to send all your blood to your muscles and to your heart and to your lungs to breathe and pump oxygen and make those muscles work, because that's what you're doing at that time, is running. And also you're going to send some to your sweat glands probably to help you sweat. But when it's time to rest, you're done with the race and you're laying around, now it's time to send blood to your intestines. And so now our body opens up the blood vessels, go into our intestines, sends more blood to them, allows us to actually digest our food, sends more muscles to the muscles of the stomach, and allows it to chomp up the food, and so forth. That's also when we start thinking about having sex. You don't think about having sex when the tiger walks in the room. I'm sorry, you just don't. All right, neurons are the basic functional unit of the nervous system. The neuron structure is really simple. I'm not going to make a big deal of it. I think there's a couple of videos on the D2L that you can look at if you want. But basically, a nerve cell looks like this. This is kind of a schematic. This is the nerve cell body down here at this end. The fluid in it is called the perikaryon. The nucleus is called the nucleus. And on the outside of the cell body, notice there's all these little branches, and these are dendrites. Dendrites are what receive the information. So all the information coming into this cell comes in through these dendrites from other nerve cells coming and hooking onto them here, sending information in. Then the nerve cell kind of processes that information, decides whether it's important enough, the stimuli that are coming in, for it to send the message on downstream. And this part of the axon called the axon hillock, thank you. The axon hillock decides whether this should be sent along, and if so, it sends a message down the axon to these teledendria things, and then these things hook on to either a muscle, or they hook onto a gland, or they hook onto a bunch of other nerve cells like this, and pass the message along. This axon can be as long as 6 feet long. In other words, if I step on a nail, okay, that nail has got a nerve cell in it that reacts to pain, and it sends a message up all the way up to my spinal cord, this long, and it stimulates another cell in my spinal cord that then sends a message back to my foot that says, "Ooh, lift your foot up off that nail because that hurts, damn it." That's a reflex, okay? And then it also sends a message on up to my brain so that about half a second later, my brain says, "Whoa, I just stepped on a nail." And luckily, I've already stepped off of it because of the reflex. The brain didn't participate in you lifting your foot up off of it. It did that automatically. That's called a reflex, okay? It's out of your own control. So here's another example of that. We're a little closer up. There's little missile bodies, which are RER, rough endoplasmic reticulum, and free ribosomes. We covered this back in the cell chapter, but you might want to review it here. Mitochondria, This is the axon hillock area. This is the area that decides whether the message is going to be sent on into the initial segment of the axon, and then once it starts, it goes all the way down the axon. Golgi apparatus live in here, neurofilaments live in here, a nucleus lives in here with a nucleolus. These are all the dendrites around the outside. Nerves communicate with other nerves by through the nervous system, and if you go back, every time that one nerve touches onto another nerve, there is what's called a synapse. A synapse is a connection, a neural connection from one nerve cell to another. And the way this synapse works in general is it's not a physical connection. They don't actually touch. What they do is the first nerve releases a substance called a neurotransmitter. Probably heard that word before on TV and all the ads all the time about a neurotransmitter, and that neurotransmitter then carries the message across to the next nerve cell to keep it going. Okay. So neurons do all this communicating stuff, and then remember the other kind of cells, which are called the neuroglia. The neuroglia are the cells that simply protect the nerve cells. They take care of them. They keep the pH normal. They keep the amount of oxygen normal. They clean up the dead cells, or they clean up any bacteria or anything that happen to be there. Basically, the neuroglia do everything that's essential to the survival and the function of the brain cells, because the brain cells, the neuron cells, are really where it's at. Think about our survival, and think how long you would survive if you were unconscious. The most important thing we can do as humans to survive is to stay conscious. That makes sense? Because if you're not conscious, you can't eat, you can't walk, you can't get away from the tiger, you can't get away from any danger, you can't get yourself out of the weather if you're laying outside on the ground and it's snowy and cold. Our brain is it. I mean, our brain is what keeps us going all the time. So we have all these neuroglia to help those brain cells out to make sure they're always working. And sure enough, the neuroglia make up about half of the volume of the nerve cells of the nervous system. Now, I haven't even told you the name of a single neuroglia yet. You notice that? I'm down here. Here they are. You need to know these cells, okay? You need to know what they are, what they sort of look like, and what they do, okay? And it's condensed very nicely into this one lovely picture, okay? And if you look at this picture, you'll notice that the neuroglia are found in either the peripheral nervous system, and the peripheral nervous system, remember, is the nerves, the peripheral nerves, and the only thing found there are some satellite cells which surround the neuron cell bodies in the ganglia. These are areas outside of the central nervous system, and they keep track of the neurotransmitter levels and so forth. There's also the Schwann cells, and this is like, this is not related to the Schwann's guy who brings the frozen food, but the Schwann cells live around every nerve cell, and they make what's called myelin. Myelin is the coating that comes on nerve cells. So if this is the nerve cell here, here's the Schwann cell that lives around it. The Schwann cell makes this white stuff, which is called myelin. Myelin is like the insulation on a wire, okay? So we've got wires running through this wall right here, and every one of them is a wire in the middle, and then it's a bunch of plastic insulation around that. And that protects the current flowing through the wire, right? It also makes the current flow faster, okay? So by insulating wires, the electrons will actually flow better and faster, and that's the way it works in nerves. so that our nerve cells that are really important to us, which is most of them, are myelinated. They have myelin around every nerve cell that makes them send messages faster. I think you have a physio X that talks about this here in the next week or so. Okay, so that's what the Schwann cells do. The Schwann cells make the myelin. Okay, inside the central nervous system, and that means this purple area is in either the brain or the spinal cord, because that's the central nervous system. Here's 4 different types of cells that are neuroglial cells. One is called the ependymal cells. The ependymal cells are kind of cool. They line the inside of the ventricles, which are the water bags inside the brain, and the water, or the cerebrospinal fluid, the fluid that's around the outside of your brain that sort of acts as a cushioning device. So when you see these guys get their heads slammed against the floor on a football game, luckily they have a helmet that's protecting their brain. Luckily they have a skull that's protecting their brain. And luckily they also have a bunch of water around their brain called the cerebrospinal fluid that tends to cushion the impact of a tough hit. See that guy the other night got knocked out, laying there on the floor, and his friends are trying to help him up. He can't get up. He's obviously out to lunch. Despite all this concussion protocol and stuff, they let him play, and he kind of staggered through 2 more plays until he fumbled and lost the game for him. But anyway, it's just kind of a sad statement that it's a rough game, and these guys get beat up really badly, and their brains take the worst of it. There's a movie coming out this next month about the concussion deal with the NFL and how for many years the NFL suppressed all the information, trying to not look bad, and all these guys that were getting Alzheimer's at early ages and going crazy and getting dementia and dying and killing themselves was not related to their chronic head injuries. So we're going to hear the whole story, I think, or at least another side of the story. Anyway, that's what the ependymal cells do. They make this fluid, and they monitor this cerebrospinal fluid. Another one is called the microglia. The microglia are they're smaller, that's why they're called microglia, and they run around and pick up cell debris, waste products. They eat up dead cells or dying cells and so forth. Then there's the astrocytes. The astrocytes do a number of wonderful things for you, one of which is they maintain the blood-brain barrier. Blood-brain barrier. I'll talk more about that later, I think. They control the gas concentrations, and they pick up and recycle neurotransmitters. Unfortunately, one of the things the astrocytes do is they form scar tissue after injury. And remember, I just told you that sometimes one cell might have 20,000 other cells attaching to it in this complicated thing we call a brain up here. Well, imagine that if you form some scar in your brain, say you got a really bad injury, you did a little bit of bleeding, even just a few drops of blood into your brain, and then you form scar tissue, no longer can all those connections go through and connect up properly. So therefore, all those memories that had to do with that particular area of your brain are now gone. Welcome to dementia. Welcome to old age. Got it. All the time, we all are forgetting things. Well, I'm forgetting more than the average Joe these days, I think. Astrocytes, that's them. Last one is called the oligodendrocytes. The oligodendrocytes are the cells that myelinate the axons that live in the CNS. So in many ways, they're like the Schwann cells, but remember the Schwann cells are on the peripheral nerves. The oligodendrocytes are in the brain. Oligodendrocytes also kind of provide a structural framework for the brain, kind of hold it in position, hold the cells in position. Please remember that the human brain is nothing like what you will see in the lab over there. The human brain is more like Jell-O, soft Jell-O. That's just the consistency of it. What you see in the lab has been soaking in formalin for at least 4 months to get stiff and hold itself in position so that we can slice it up and cut it and look at it, things like that. Okay, and it's funny how some things come back from your childhood. When I was a kid, I went to see Ford's Theater in Washington, D.C. And Ford's Theater was where Abraham Lincoln was assassinated. And the story, the history is that he was shot in the back of the head, point blank range, by John Wilkes Booth. And then everybody came up there to find Abraham Lincoln, and a doctor looked at him, stuck his finger in the hole in his head back here, and declared this was a mortal wound. And that was pretty common back in those days because they couldn't fix much of anything. There wasn't any such thing as neurosurgery or even anesthesia back in those days. And so they all picked him up and they carried him across the street and they put him in this boarding house, which is directly across the street from the theater. They put him in the biggest bed they had, which was a double bed, and they had to lay him crosswise because he was a really tall guy. And the stories that I read as a child about that were that, and through the night, Abraham Lincoln's brain oozed out onto the bed. And there was a big pile of his brain tissue there by morning when he died at 7:23 in the morning, I think. They found his heart stopped and they declared him dead. The point is that the blood, that's what the brain is like. It's like soft Jell-O. And if you make a hole in the head, it's just going to come oozing out and just fall apart. It's also why you never, ever shake a baby, because it's like you're shaking a bowl of Jell-O. And when you shake it, you're going to tear little pieces of it. That Jell-O is going to come apart, and everywhere that it comes apart is one more area of that baby's brain that will never work right. And that's why so many babies die from being shaken, because their brain is literally ripped apart into thousands of pieces. I have pictures of that. I'll show you before we get through all of this, I think. So let's go through them again. Ependymal cells are the ones that form the epithelial layer called the ependyma. They line the central canal of the spinal cord, which is the little hole that runs down the length of the spinal cord, and they also line the ventricles of the brain. They have cilia and microvilli that circulate the CSF and keep it moving. These ependymal cells also monitor the CSF and can correct things like pH and oxygen levels and so forth. And they also contain stem cells, so that if a bunch of ependymal cells get wiped out, they can replace themselves. You got to remember now that brain cells can't be replaced. So you probably turn to one of your friends sometime during your life and say, "Wow, we really got smashed last night. I think I killed off about 100,000 brain cells." You did. You killed off 100,000 brain cells that night you got really drunk, and they will never come back to you. Now, you're lucky because you have a few billion, so you'll probably be okay, but just keep that in mind. Here's a picture of the spinal cord cut transversely here, and there's a little hole that runs down the middle of it. We'll learn about more. It's called the central canal, and it is completely lined with ependymal cells. So these are all ependymal cells around the edges of it, and they control that fluid. That fluid also runs around the outside of the spinal cord out here to kind of support it and give it protection as well. Second one is the astrocytes. The astrocytes play a role in maintaining the blood-brain barrier. Let me digress here just for a minute, talk about the blood-brain barrier, because as I told you, the most important thing to human life is staying conscious and keeping our brain working right. In order to keep our brain working right, we have to control the level of all the chemicals that are in it. Now, sometimes we do things to alter that chemical stuff in there on purpose. We might drink too much beer. That puts too much alcohol into our brain, and it makes us not think right. And everybody knows that, right? So that's because alcohol can cross the blood-brain barrier. Alcohol can get out of your stomach and into your bloodstream, and it can cross the blood-brain barrier when that blood circulates up through your brain. It can get out of the bloodstream and get into the brain tissues themselves and make you drunk. Okay? Some chemicals can cross into the blood-brain barrier, others can't. The blood-brain barrier is this kind of magical and hard to describe system which allows our body to try really hard to keep all the chemicals that are in the brain cells themselves at a normal level by letting some things get in and not letting other things get out. Not letting other things get in. The best example I can tell you for that is antibiotics. Back in the day, when I was practicing medicine, we had certain antibiotics that we knew could cross the blood-brain barrier. Some of the drugs that we used to fight infection could cross the blood-brain barrier and get into the brain. So if you were treating somebody for meningitis, you would sure want to use one of those, right? Because you want it to get into the brain to kill the bacteria to cure the infection. Whereas there were other antibiotics that did not get into the brain at all. And early on, what doctors would do, if they had an infection that they only had, say, one antibiotic that worked against that particular bacteria back in the day, they would actually inject the medicine straight into the brain. Whew. How's that for a thought? Back in the 60s and 70s. Somebody... But that's the only way there was to get those certain antibiotics in there to fight those certain infections. Now, nowadays, we've been able to perfect all kinds of drugs that can pass into the blood-brain barrier, so that's not an issue anymore. As far as I know, we can modify every antibiotic to allow it to pass through the blood-brain barrier and get into the brain. Perfect example of the blood-brain barrier is with blood sugar. Normal blood sugar level is about 100. If you're diabetic, it's not unusual for your blood sugar to go up to 400 or 500, and you don't even feel any different. And in fact, and it's not until your blood sugar gets up to 1600 or so, which is when you're in real trouble, actually. But then you notice it because you kind of get sleepy and you're going into diabetic coma, that sort of thing. Because your body controls, even though the blood sugar out here in your arm might be 1400, say, in your brain, it's still exactly 100. It's exactly normal. Because the blood-brain barrier can so carefully control the amount of sugar that gets in and the amount of sugar that gets out, that your blood-brain barrier can keep your blood sugar in your brain cells itself normal. Pretty magic, huh? So these astrocytes maintain this. They basically isolate the central nervous system from all these other metabolic issues. They create a little three-dimensional framework. They repair damaged tissue. They guide neuron development, and they control the interstitial environment. I think I've made that pretty clear. The other one is oligodendrocytes. Remember I said that oligodendrocytes make myelin sheaths, and these myelin sheaths are what insulate the nerve cells one from another and also speed up the transmission of the signals through there. Myelination is something we will spend some time talking about, but myelination is, like I said, it is the coating on the outside of the wires. It's the coating on the outside of the axons. It increases the speed that the action potentials can travel through it. By being insulated, they flow a lot faster. It insulates them so they're not sending the sparks off the side to other things. It also makes all the nerves appear white. So you probably heard the term gray matter and white matter in the brain. You're going to hear a lot about it here in the near future. But just know that if it's white, The reason it's white is because it's got a lot of myelin. The white matter of the brain is almost always just a whole bunch of nerve fibers going in the same direction, and all of them are myelinated, so it looks white. The spinal cord will have areas that look white and areas that look gray. The gray areas look gray because they have all the cell bodies, the nuclei, and center part of the cells in them. There are things called nodes and internodes, which are basically areas where the internodes are where it's myelinated, and then there's also these nodes, which I think you've already heard about, called the nodes of Ranvier, which is where the myelin is broken into segments, so to speak, and there's a little gap where the axons may branch there at those areas, but it's also where it slows down the nerve transmission a little bit. That's actually where the nerve transmission passes to in myelinated nerves. I mentioned this already because I forgot it was on this slide. I always get ahead of myself. Y'all figured that out. White matter is where there's it's regions in the brain, in the CNS, that have lots of myelinated nerves. And gray matter are areas that are either unmyelinated, but in general what it really means is the gray matter is where the nerve cell bodies are located, where the nuclei are located. Another type of neuroglia is the microglia, the little ones. They can migrate through the neural tissue, cleaning up debris, waste products, pathogens, bugs, bacteria, all that jazz. Here's, thank you. Here's a schematic. It's really kind of cheesy, but it shows the central canal out here, which is the water flowing through the spinal cord, for example, and it's lined by a bunch of ependymal cells. And then out in here, there are a bunch of. This is a neuron cell here, a brain cell, the blue one here, and notice that it has a whole bunch of telodendria attached onto it. And as I said before, you can have as many as 200,000 different nerve cells putting dendrites on one nerve cell and contributing information to that one nerve cell before it decides whether it should send the message on down its axon to the next one. Here's a microglial cell, one of those picks up the garbage. Here is these are oligodendrocytes here, right? The ones that make the myelin inside the nervous system. You can see it making myelin around this branch right here, and so forth. I'll just leave it at that. What is a ganglia? A ganglia is an area of a bunch of nerve cell bodies that are found in the peripheral nervous system. They're not in the brain, they're not in the spinal cord. We have lots and lots of collections of brain cells in the peripheral nervous system. We don't have very many ganglia outside. We have like a ganglia that lives on the outside of the heart. And axons carry signals there, and then it sends signals to the heart muscle itself to either slow it up or slow it down. Satellite cells surround those ganglia. I'll show you examples of that when we get to the spinal cord, just around the outside of the spinal cord. Schwann cells are the ones that form the myelin sheath around the peripheral nerves. It's one Schwann cell for each segment of the axon. So there may be many Schwann cells surrounding just one nerve, because remember, an axon can be this long, and it has a whole bunch of Schwann cells along the side of it there. Here's kind of a close-up of what that looks like. You'll see this in the lab, by the way. You should look at them under the microscope. Here's a bunch of axons right here, these little blue structures. And notice there's a Schwann cell, and the Schwann cell is making myelin. It's this yellow stuff that completely surrounds those nerve cells. So one of the things that the Schwann cells do is they make this myelin, But in the process, they also make these sort of little tubes that allow the nerve cell to follow through them. And the reason that's important is if you ever cut a nerve, okay? So if you cut a nerve right here in your arm, okay, there's a little nerve running underneath this skin there that carries the messages down to this whole part of your hand, for example. And if you cut this nerve, you're going to probably go into the hospital, and the doctor's going to do a little pin test, and he's going to test to see where you can feel and where you can't feel. And based on where you can feel and where you can't feel, he's going to be able to say, I know my anatomy. I remember A and P, and therefore you cut your radial nerve. Now I know that the radial nerve goes this way and goes around there like that, so we can go find it. So you go in there surgically, and you make an incision, and you dissect down through there until you find the nerve where it's been cut, and you find the two ends. And you take the two ends, and you might cut them off a little bit more if it's kind of rough on the end to make it nice and sharp. And then you take in little stitches, and you take that nerve, and you sew it right back together. Okay? Now, the axon from the place where it was cut on down is all dead. I guess this way, from that way, it's all dead. Why? Because the nucleus and the mitochondria and everything that supply that nerve live up there, right? So you sew them back together, and you try to get them lined up just perfect, and you come out of surgery, and the patient says, well, doctor, it doesn't feel any different. I'm still numb. And you say, yep, you are. And it's going to be that way for a good long while. Because nerves only grow about 1 millimeter a day. So even though you repaired it here, for you to get feeling back in your hand, it's got to grow this many millimeters. Well, you know, there's 10 millimeters to a centimeter, and this is about 30 centimeters. So 30 times 10 is 300 days, is that right? Not that long. Anyway, it's going to take a long time, and so you just tell the patient, listen, yeah, we put your nerve back together, we can't guarantee it that it will grow back, but you got to be patient because it will come on very slowly. And in fact, when it finally does get growing back, it'll feel tingly, and then it'll feel probably hypersensitive, and then it'll come back to normal. So the one good thing about these Schwann cells is they do make these little tubes, and so as those nerve cells start to regrow and extend their axons back out there, I hope that's not mine. Anyway, it serves as a pathway for the nerve cells to grow back through. Yeah. Yeah.
What do they look like? Are they, because in the diagrams, like in the dissection picture that have, they're kind of white.
They do look white, and in the cats, they're pretty small. A nerve, like for example here, that goes down into your hand in a human would actually be probably 3 millimeters in diameter. They're big enough to see. Up in the brachial plexus up here, we'll talk about that. They're 5 to 6 millimeters in size. They're pretty good size. So anyway, I think I've said this. Remember that the neurons do all the thinking and do all the brain stuff. The neuroglia simply support them. This is that business I was just talking to you about, Wallerian degeneration. If you cut your nerve, everything distal to that point dies. The reason that we don't grow back brain cells in our brain and don't make all these reconnections is because scar tissue is formed, and when the scar tissue reforms, their reconnections can't occur very well. Okay, what happens in nerve cells? Because I've kind of talked to you about it as we've gone through this far. What really goes on in nerve cells? And let me just start by talking about this. How many of you guys sitting in this room for the last hour have been listening to the fan in that projector? How many of you can even hear it? Now that I mentioned it, I bet you can hear it, right?
Heard it.
You have heard it.
Just choose not to listen to it.
It bugs you.
Or I wrote focus.
Yeah. Okay, now, all right, that's good, because that's one of those sounds that some people hear it and some people don't. It's the same, it's constant. Our brain tends to suppress it because we're interested in what else is going on. All right, let me ask you this question. How many of you in the last 15 minutes have felt the weight of your shirt on your left shoulder? How many of you have felt the weight of your shirt on your left shoulder? Huh? Nobody has, right? Why? But yet you can. You know, if something touches you on the left shoulder, or if you lift up your shirt, you can feel it without any trouble. Why did that happen? Why didn't you think about it? Because, good God, think about it. If you thought about everything that was coming into your body all the time, all the sensation, all of the signals that your body was sending you, you wouldn't be able to think about anything else, right? So we have a way of filtering out huge amounts of information that's coming into us all the time. We call this information processing. Information processing means that we sort of integrate the stimuli. In other words, everything that comes in doesn't necessarily get passed along and go up to our brain for us to think about. And the things that we tend to suppress are things that aren't painful, like the sound of that thing up there. That's not really painful, is it? They're constant. They don't change much, same as the weight of the shirt. And so our brain says, well, that's really pretty useless information. I don't have to think about that, and it doesn't. It processes it out, and that happens at many levels between your ear and your brain. Same as between the feeling in your shoulder and your brain. So this is kind of a quick and dirty overview of what happens in brain cells. We get inputs coming into this nerve cell over here. Here's the nerve cell body. And this nerve cell receives, it sits normally in what's called a resting potential. It has a certain electric charge on it, okay? And that is a resting potential when nothing's really stimulating it. And then it starts receiving all these inputs from all these dendrites, and some of those inputs then cause the potential to change a little bit. And these are called graded potentials. And if you get enough graded potentials that all say, hey, this is important, you need to think about this, basically the way that does that is it causes a greater change in the resting potential, turns it into a potential that is higher, and if there's enough input that signals it hard enough, then this part of the cell right here, called the axon hillock, produces an action potential. Boom. It says, well, must be important. Let's send an action potential on down to the next set of cells. So here's the next cell down here. Once an action potential starts, it goes, and action potentials are all or none, kind of like muscle contractions, you remember? So if the action potential starts, it goes all the way down to the other end. It hits these synapses down here. Remember the synapses which release the neurotransmitter? The neurotransmitter then goes across the gap here to the next cell, and then that cell has to decide whether it wants to send the information on or not, whether it's important enough, whether it's strong enough signal. That every time we do that, every time we decide whether there's enough here to carry on or not, we call that information processing. In other words, these nerve cells are deciding whether this is important enough. And the way they're deciding is by how much did the resting potential, the charge on the wall of the membrane of these nerve cells, how much did it change? If it changes enough, boom, it sends an action potential on to the next one. If it doesn't, it just sort of dies there. The transmembrane potential is based on several things. What is it that causes there to be a charge between the inside of a cell membrane and the outside of the cell membrane? That's what the transmembrane potential is. The fact that there's a different charge. Now, it's measured in millivolts. It's very small, but it's there nonetheless. And it depends on the concentration of ions. These are both cations, right? They're positive. Sodium and potassium are really big ones. And sodium and potassium can move in and out of the cell through different channels. Some of them are selectively and allow sodium in. Some of them are selective and allow potassium in. And as they do that, and there are pumps in the membrane. You remember studying about those back in 112? So that we tend to pump sodium out and we tend to pump potassium in. Okay? Because the level of potassium inside of cells is quite high, whereas the level of sodium on the outside of cells is quite high. Okay? There's also another thing that contributes to this, and that is the proteins that are in the cell. There's obviously a lot more proteins in cytoplasm than there are outside of the cell, in the interstitial fluid or in the extracellular fluid. Okay? The combination of all these charges, and because of these pumps, leads to this number here of a resting potential of about minus 70 millivolts. That's caused by a chemical gradient and an electrical gradient, which results in this potential difference. If we can look at the membrane up really close, please notice that it is more negative on the inside. Now we call that, it's more negative on the inside, and so we call that a minus 70 resting potential. We could just as easily call it a plus 70. Somebody just had to decide which is going to be plus or negative. Okay, so we measure basically based on the inside charge, and the inside is more negative, and so therefore we call this the negative 70 is our resting potential. Now what makes it that way? Well, one of the things is that there's a whole lot more proteins inside. Here's the proteins, these little blue blobs down here, and you notice there's none of them out here, and they're all negative, so that makes the inside more negative. Okay. The other thing is that we have this sodium-potassium exchange pump in the membrane that is causing, it's an active transport system, which means it is burning ATP, right? Active versus passive. Passive and diffusion don't require any ATP, but this is one of those pumps that does. And this little pump is constantly pumping potassium in and pumping sodium out. Sodiums are the blues, potassiums are these brownish colored, okay? So we end up with a much higher concentration of potassium on the inside. See how many brown pluses there are on the inside? And we're ending up with a much higher concentration of sodium on the outside. And that's the blue ones. And look how many more of the blue ones there are here on the outside. Interesting. Now also notice that you got a bunch of chlorides out here, because you can't have sodium and not have some anion there with it, right? And so usually these are chloride ions, which you see illustrated as green and bigger. All right. Now there are leak channels as well. Remember, leak channels, they just are open and leak all the time. So despite the fact that this pump is working like hell to pump all that sodium out and pump that potassium in, you've got these leak channels that are just allowing the potassium to leak out, and that are also just allowing the sodium to leak in and making this concentration go away. But that's what life is all about, okay? That's the difference between something being alive and it not being alive, is the fact that there's enough mitochondria around making enough ATP to keep these pumps going, to keep cells alive. Because cells don't survive if they don't have their pumps working, okay? And that's why this cell is a pretty standard cell. I think a nerve cell in this case. But really the only difference between nerve cells and other cells is the fact that nerve cells are sort of made for this. They're sort of made to maintain this electric charge difference. Okay? Because nerve cells are all about sparks. They're all about these electric charges. That's what nerve cells are about. Sessions are getting really weird. All right. In the course of these, here's 3 classes of gated channels. Okay. Now, I told you that there are these gated channels, and some of them work only in the presence of certain chemicals, like acetylcholine. Remember back when we were studying muscle connections and the neuromuscular junction, the action potential came down the nerve, and it caused those vesicles to open up and release the acetylcholine out into the gap, and the acetylcholine went across the gap and opened up some channels on the other side, remember? Okay. Well, that's the same thing that goes on here. I mean, that's actually part of what we were talking about before, is that there are these chemically gated channels. And so the only reason these channels open is because there's a chemical present, and the chemical we've been talking about is acetylcholine. Okay? We also have channels that open based on the voltage, so that, you remember, going back a little step from what we were just talking about, you remember how that action potential came down the nerve, and when it got to the end of the nerve, to the telodendria, it caused a bunch of sodium to rush in. It changed the gates, and it caused sodium to rush in, which then caused the exostosis to occur, to open up all those little packets of acetylcholine. So those were gates that were controlled by voltage. Okay? This is pretty characteristic of these excitable membranes like neurons and skeletal muscle, sarcolemmas, and cardiac muscle, and so forth. So keep these both in mind. Some of them are opened by voltage coming in the form of a spark. Some of them are controlled by chemicals. Usually it's a spark that causes the chemicals to be released. And then the other one is called mechanically gated channels. Now let's think about that for a minute. Let's think about a gate. Okay? Here's a gate. Okay, this is the 2 sides of the gate, and the gate is in between. Put your finger right in between, right in there. She's got her finger in the gate. Okay? Now, things are trying to get into the cell and change potentials around. And if I could pull these gates apart, then it would be a whole lot easier, and she could get 3 fingers in there and a whole bunch of chemicals could flow in, right? And if it wasn't being pulled apart, it was staying tight together, then nothing could get in, okay? Now, where might that be helpful in your body? Well, sounds weird because I'm going to bring something down from the big level to a microscopic level, but let's consider the skin on the back of my elbow, okay? So I have feeling fibers in there, and how do I know whether my elbow is straight or whether it's bent? Without looking, how do I know whether my elbow is straight or it is bent? Well, one of the ways it knows several ways, because it has nerve inside the joint and around the muscles and so forth, but it also has nerve fibers in the skin, for example, okay? In other words, when I have my eyes closed and when I bend my elbow like that, I get a signal to my brain that says, hey, pal, your elbow is bent. And the reason you know that is because your skin is being stretched across the end of your elbow there, okay? So let's think about this as being my skin here, and it's got all these gates in it, and I bend my elbow, which stretches my skin, which mechanically opens up the gates, and allows the chemicals to go inside the cell, which changes the impulse, which then sends a message to my brain that says, hey, skin of your elbow is being stretched. Okay? Those are what are called mechanically gated channels. In other words, they actually respond to distortion of the membranes themselves. Mechanically, they're being pulled apart. Get it? They're found in a lot of sensory receptors like touch, pressure, vibration. Same thing, you want to feel vibration in your arm, take a tuning fork and push it down on there, and it's going to go wah, 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 and your brain is going to figure that out because of these mechanically gated channels that are going to open and close, open and close, open and close, and send you the message. So here are representations of these three things. You need to understand the three different kinds of gates. Chemically gated channel, nothing can get through up here, but then when the correct chemical transmitter arrives, for example, acetylcholine comes in here, binds in there, another one comes over here, binds in there, they open up and allow ions to go inside. Okay? So it was the chemical, the red dots here, the acetylcholine, that caused this to open and allow the gates to open. Next one, voltage-gated channels. Here's the voltage, it's minus 70. Down through the axon, down through the membrane comes this action potential, which changes the voltage to minus 60, for example. And when that happens, it causes these gates to fly open. When these gates fly open under the influence of the voltage, that allows ions to flow inside, which then changes the voltage, changes it here to, what is it, plus 30? Plus 30. And when it gets far enough, then the gates start to close back again. But these gates are strictly under the control of voltage. Okay, so these are voltage-gated channels. Finally, here's the mechanical ones I was showing you. They're closed because this membrane here, the skin or whatever it is not under tension, but then when you stretch it due to applied pressure, it shows here, you push down on it, stretch them apart, opens the gates, in comes the chemicals, changes and sends off the messages. Once the stimulus is gone, everything comes back to normal. Okay. Hyperpolarization occurs at the end. It always overshoots, is the point of the matter, is all of these things, they kind of overshoot. And so I'll let you just read about hyperpolarization. Here's a kind of the graph of it. If normal is minus 70, we depolarize it. It turns it up to about minus 60. Then it can repolarize, come back to resting potential. But sometimes it will hyperpolarize. Here's a chemical stimulus applied. It hyperpolarizes, goes this way instead, but it eventually also can return back to resting potential. So hyperpolarization just becomes, it's just saying it becomes more negative. It was normally negative about minus 70. Repolarization becomes less polar. Hyperpolarization becomes more polar. Okay, I'm going to stop right here on graded potentials. Yeah, I'll just stop here. Look at here. 46 out of 90. Wow, we got almost halfway through, didn't we? Just right at halfway. All right, we'll finish that up on Tuesday, like I said. Any questions? Okay. We will meet in lab in a little bit. Okay, we're going to talk about the rest of the nerve cell chapter here, and then we're going to go, and that'll be it. Then we'll go into spinal cord next time. Okay, graded potentials. I think there's a couple of terms you really need to understand. And, I would just say that graded potential means that it's not necessarily a potential that's strong enough to send off an action potential. So indeed, in the brain, where I've mentioned there are cells that have contact with 200,000 other cells, they may all be putting in information, and maybe only every so often is there enough enough potential change sent there to make an action potential actually fire off. So think of graded potentials as subtotal action potentials. Okay, and you have to add several graded potentials together sometimes in order to have the cell send off an action potential. So here you go. What are the effects of them? Well, they can be a lot of different effects from graded potentials. For example, sending cell dendrites and cell bodies telling the cell to make more protein of one type or another, or to cause it to exocytosis, the glandular secretions. Obviously, at the motor end plate, the action potential causes the release of ACH, acetylcholine. So, the thing you need to understand, and I would tell you to review this in your book, and I think I have some videos, some brief videos I'll show you over in lab off of the video, that disc that I have. What is an action potential? Well, an action potential is a potential that once it fires, it is going to go the length of the axon. It's going to go all the way down, and it's going to affect the entire excitable membrane. Now, if it happens to be a motor neuron axon and it's going down there, when it gets to the motor end plate, it's going to cause the release of the acetylcholine. The acetylcholine is going to go across the gap, the synaptic cleft, and it's going to fire off muscle contraction. So, let's talk about these a little bit. So, the whole thing is sort of process. When we say process, I like you to think of nerve cell of action potential processing sort of like thinking. In other words, the cell membranes assess all of these incoming graded potentials and then decide whether to fire off an action potential down the axon. That happens at the axon hillock, remember? That has to be big enough, 10 to 15 millivolts, in order to change the resting potential enough to actually fire off that action potential. But once that action potential fires off, it's going all the way to the end of the nerve, which can be this far, right? Think about this. When the stimulus actually exceeds the threshold amount and it fires off that action potential, the action potential is going to be exactly the same. It's going to be the same size. It's going to be the same duration as all the other axon potentials. No matter how large the stimulus coming into that nerve cell through all those dendrites, it's only going to send out one action potential, right? So, we have this principle called the all or none principle. And the all or none principle says that if an action potential is fired off by the axon and sent down the axon, it's all or none. It either does it or it doesn't. In other words, it either reached stimulus threshold or it didn't. Now, how does this happen? Well, I'm going to run through these slides, but to be quite honest with you, I think you got to go over this on your own using the same pictures that are in your book and go through it step by step, and I think it becomes quite easily understandable. So in the resting state, as we've talked before, the normal charge is minus 70, which means it's minus. More minuses are on the inside of the cell than they are on the outside. This is the cell membrane here. These are a couple of gates. Okay? So on the outside, you see a bunch of pluses. You see hardly any negatives. On the inside, you see a lot of negatives. Remember, a lot of those negatives are from proteins. Proteins that are concentrated inside the cell often are always carry a negative charge. So anyway, the charges are more negative on the inside to the tune of about minus 70 millivolts. And so really there's 4 steps in the generation of an action potential. One is depolarization down to the threshold. And then once we get depolarized down to that level, the sodium channels get open. What happens when the sodium channels get opened? Well, the sodium channels rush in. And you got to remember, sodium is always more concentrated on the outside of the cell. Potassium is more concentrated on the inside of the cell. Okay? So, once you open the sodium channels, the sodium ions are going to rush in. Now, those are plus ions, positive cations, coming from the outside to the inside, and that is going to change the membrane from negative to positive. Okay, it's going to change it from minus 70 up into the positive range. So here's what happened. Here comes this electric charge down the membrane. It's going to open this gate right here, and these sodiums are going to pour in, as illustrated in the next picture right here. So now with all these pluses running inside here, it's changed the microvoltage from minus 70 up to plus 10. So now it's actually more plus on the inside of the cell than it is on the outside of the cell. As it does that, as the action potential passes on down the membrane, these sodium channels eventually close, and it activates some potassium channels. We have specific channels for both sodium and for potassium. Well, think about this. If you open potassium channels, which way is the flow going to go? It's going to go out, because remember, potassium is much more concentrated inside than outside. So following the old diffusion rule, and osmosis rule, whatever you want to call it, out it goes. Okay? So, but going back to that, once these sodium channels become inactivated, the potassium channels become activated. So here we go. The action potential has now passed. Basically, it's going on down the membrane. Now the sodium channels are snapping shut. Potassium channels are snapping open. Potassium is rushing out. And look what's happening to it got as much as plus 30, and now it's going to all come back to normal. The potassium channels are going to now close, and the membrane is going to reestablish itself back to its normal resting potential of about minus 70. Now, how does it do that? How does it go back to minus 70 when it was just at plus 30? No. What makes this concentration difference to start with? No. The pumps. Remember the active pumps that pump sodium out and that pump potassium in. So once this whole thing is passed, those pumps, which aren't illustrated here, are going to click back into gear and start pumping the sodium out, pumping the potassium back in, okay, to reestablish this high level of concentration of potassium on the inside, high level of sodium on the outside. Now, the reason it's important for you to get this concept, especially later, is when we start studying fluids and balances and salts and kidney function and all this jazz. All of these factors will become very important. So it's like most everything in A&P. If you learn it right the first time, you got it. Okay? So, here we go. Step 4, the return to normal permeability. The sodium channels are closed. The potassium channel is now closing, and it's going to go back to minus 70 because those pumps are going to reestablish it. All right, a couple of terms you need to understand. What is the time period from the beginning of the action potential until it returns? Well, believe me, it's very short. I think it's 3/1000th of a second. No, 7/1000ths of a second. You might want to check me on that number. I'm not going to ask you the number, but just know, so you know it's very short. what is the absolute refractory period? And what the absolute refractory period means, it's the period of time where no matter how much more input you get, it will not fire off another action potential. In other words, it's absolutely refractory to another stimulus. Okay? And that lasts basically for just as long as the sodium channels are open. It's a pretty short period of time. But following that, there is a relative refractory period where, the cell doesn't really want to fire off again, but if you hit it with enough, a very large stimulus, you can initiate another action potential. Sodium potassium pump, I've already discussed or mentioned at least. It's the one that's going to work all the time without action potentials. It's going to keep working in order to maintain these concentration gradients of sodium and potassium. This is an active transport pump. It takes energy, i.e., it takes ATP. And so, in other words, and you ought to look over this in the book as well, it takes 1 ATP to pump every 2 potassiums in and to pump every 3 sodiums out. It's a 3-to-2 exchange ratio. What happens if you don't have ATP? What happens if you don't have any energy to power this whole thing? The neurons don't work. You're dead. Certainly your muscles don't work. Certainly your brain doesn't work. You're probably dead. Here's a chart of this whole thing going from the -70 up here to the +30, coming back down here to even lower, maybe -100 down here. Notice from the, so this is the resting potential. It gets these graded stimuli, and they change it up to about -60, and that's threshold. Once it hits threshold, boom, the channel's open, and boom, it goes all the way up here to +30, and then it gradually starts repolarizing back down through here. Now notice, until it gets down to about this level right here, or -40 or so, it's in what's called the absolute refractory period. And no matter how much of a shock you give it right here, it will not fire off again. Now once you get over here into the relative refractory period, if you give it a really big jolt, it will fire off again, but it takes a higher than normal stimulus to get it going. Let's talk about continuous propagation. Okay, now what we're talking about here, change thinking hats a little bit. We've now had the depolarization, and we've had it start off this action potential, sending this action potential down this nerve. How does it go down the nerve? How does it go from, let's just say we have a nerve, here's the cell body over here, okay, and there's the muscle down at that end it's going to be stimulated. And we know that once an action potential starts, it's going to go all the way, okay? How does it do that? That's what we're talking about right now. There's 2 ways that it happens. The first one is called continuous propagation. And let me try to summarize this in a simple display. If it's got to go from here to there and it's continuous propagation, it's going to depolarize this little area, and then it's going to depolarize this little area, and then it's going to depolarize this little area, and then this little area, and this little area. It's going to work its way all the way down. It's kind of a slow process, okay? So basically, it's a very local current that involves it, and I think this is probably a pretty decent picture. Notice that as this area starts depolarizing, then the charge changes here to +30, and then this is -70, and then as these electrons, these cations, influence this, and as the action potential moves down, it then causes depolarization of this segment of the membrane, and then this segment, and this segment. And you notice in order to make that happen, these ions have to basically move right along with it. action potentials can go in one direction at about 1 meter per second. Okay, now that may seem fast to you, but that's really pretty slow. Okay, here's a meter, 1 second. That's about how fast it's moving, 1 meter per second. Now, let's talk about the other form of propagation of an action potential down the length of an axon, and that's called saltatory propagation. And saltatory propagation only occurs in myelinated axons. Now remember myelin? Myelin is that white insulation that's on the outside, and it has breaks every so often in it. Maybe like this. There's a little break, and we call those breaks nodes of Ranvier. That's right. Now, the way it works in this type of propagation of the signal from our cell here to our end of our cell, or end of our axon down there, is it jumps from node to node. So instead of going this little piece and this little piece and this little piece, and all the ions being local here, with myelin, it can't. It's insulated. It can't move ions in and out between here and there to the next node. So what it does is when it gets here, it causes influence down here. The next place that ions can get in and you can open gates is down here at the next node. So basically the action potential goes boom, to boom, to boom, to boom. It travels very quickly. It jumps from node to node. I think it says that right here. Local current jumps from node to node. And the depolarization doesn't occur every millimeter like it did in the slow form of continuous propagation. It only depolarizes at the nodes. So it's a hopping down. It's much, much faster. So I think this is a pretty good little area to understand. Here's the initial segment of the nerve. Here's the point of the nerve. And notice there's several nodes. And it's really only at these nodes that there is this ions moving in and out. Because this, from here to here, it's all blocked. It's all myelinated. It's all insulated from here to here. So the next place that even can have anything happen is down at this node, then to this node, and so forth. So saltatory propagation only occurs along a myelinated axon, and it's very, very fast. And it also relates to the other thing that controls conduction speed, and that's what we're talking about here, is nerve conduction velocity. How fast is this transmission going down from here to there? And that's affected by two things. Number one, whether it's myelinated or not, because obviously if it's myelinated, it's going to do saltatory. And the second of is the size of the nerve, the axon itself. Bigger axons carry electricity a lot faster. I wired a house one time, and you have this electric box here, and you have to run wires to all the plugs, and plugs don't usually carry too much current, so it's kind of a small wire that you use, maybe 14 gauge wire, and the wires are, about as big as a pencil lead inside their little insulated covering. But then I had to wire in an oven, the kitchen oven. And the oven requires a huge amount of electricity. When it turns on, the elements burn lots of electricity, so you have to have more ability to send more electricity. And so what you do is you wire that with a very big wire, about 24 gauge, 28 gauge, much larger wire. Maybe I'm saying that backwards. Maybe it's 8 gauge because it's bigger. But anyway, bigger wires carry more electricity and they carry it faster. Same as happens here. The larger the diameter, the lower the resistance, the faster the charge. And because of this, we group our axons into 3 groups, A, B, and C. I think that's kind of a silly naming system, but nonetheless, think of it like this. If they're bigger, they transmit faster, right? So know that. When the diameter is bigger, the transmission is faster. When they're myelinated, does that make them faster or slower? That makes them faster. The speed of the action potentials that goes down those wires, which is it fastest in? It's fastest in the ones that are myelinated, and it's fastest in the ones that are big. So big axons that are myelinated are your fastest conductors of all. Okay? So, now, this goes to this ABC thing, but I think if you know those two concepts, you got it made. Bigger is faster, myelinated is faster. Okay? So, high speed, type A fibers, 140 meters a second. Now, I told you that the little fibers that are non-myelinated, that use continuous propagation, 1 meter per second. Okay? Okay? These guys here, 140 meters per second, it's and it's in the cafeteria. Okay? It's in the cafeteria. It goes that much faster. Okay? Now, think about this. Why would you need fast nerves, and which ones would it matter if they were very fast? Watch this right here. I hate giving examples like this. It makes me look so stupid. I'm going to try to balance on one leg. Okay? I'm probably a little more wobbly than some of y'all would be if you stood on one leg up here and held your arms out. But why am I wobbly? Because your muscles are what? They're fatiguing. One muscle group is fatiguing, and the next one's trying to get kind of in place. What else? As I move, I'm just sending these messages up to my brain, and my brain is sending messages back to keep me in balance, right? And my brain is using all kinds of signals. It's using signals from my eyes. I am actually using my eyes to balance. Yes. And if you don't believe it, watch here. I'm balancing with my eyes open. Now, if I close my eyes, it's about all I can do to balance, honestly. We need all of our senses in order to balance, and we need to make all these changes to our muscle tensions and so forth very, very quickly. So, position, balance, touch, motor impulses. Also, eye transmission is not on there, but it's got to be very fast too. You got to be able to see things in a hurry, right? If they're flying at you. Okay, so those are the type of things that would be carried by large diameter, myelinated, type A, really fast fibers. Well, how about other things? Not quite so important. Well, let's take these guys here, type Bs. They're usually myelinated, but they have just medium diameter, so they maybe only travel about 18 meters per second. In other words, they go from this wall to that wall down there in a second. 1 second. To the other wall. Still pretty fast, but not like the one that went to the cafeteria in the same amount of time. So these are things like sensory information, like what you feel, peripheral effectors like muscles to move, to respond to those things, and so forth. Slow, unmyelinated fibers, slow diameter, very slow, the 1 meter per second ones that we talked about earlier, carrying slower information, carrying it by continuous propagation as opposed to saltatory propagation. Things like involuntary muscles. Those would be like the sphincter at the other end of your stomach, called your pyloric sphincter. It kind of, you eat a meal, your stomach fills up, and then a couple hours later it starts to open up a little bit and allow food to start passing out of your stomach. Obviously, not a thing that has to respond very quickly, it can respond very slowly. Most all of our smooth muscle is involuntary muscle. And how about gland control? Let's say I'm running and it's starting to get hot, and I say my medulla in my brain says, hey, we better start sweating here because we're getting hot. It doesn't really matter if that signal goes all that fast, does it? As long as it gets there, half a second or a second, even at those slow rates, you see, it's going to get there in a second. That's okay. Okay? Information travels quickly through as electrical signals. And this is the point I've made several times about the baby and the fact that our thoughts are really nothing more than just patterned shocks, patterns of electric shocks that are passed around between nerve cells. Most important information, vision, balance, and motor commands is carried by the large diameter myelinated axons. That's really about what I want you to know about that. I believe we've already talked about synapses because we've already talked about muscle contraction. This is kind of a little bit of a review. Just know that action potentials, which are these nerve impulses, are transmitted from a presynaptic neuron to a postsynaptic neuron or other postsynaptic cell. Now that would be the idea in the neuromuscular junction, where it's going from a nerve to a muscle cell, okay? It could also go from a nerve to a gland cell to make it secrete or whatever, okay? Nonetheless, whenever it does that, it has to cross a synapse. Synapse is nothing more than a connection between two nerve cells. Most of our nerve cells transmit information via chemical synapses. The example of the muscle we've already talked about was we release acetylcholine, it goes across the synapse and causes the stimulation on the other side. Believe me, most of our brain-to-brain cell, and there are things up here that make us think, connection from one brain cell to another brain cell is usually using acetylcholine as well. There are a few systems in our body that use just pure electrical synapses where the two cells are actually in direct contact. We're going to talk about that when we get to the heart. The heart, because it all beats together, and the muscle cells, you want them to all beat together. They just physically contract with each other. It's a direct contact thing as opposed to nerves. But for now, what I want to talk about is just the chemical synapses. Most chemical synapses are found between neurons, and they're found in all synapses between neurons and other cells, for example, the neuromuscular cells, like we talked about. The cells are not in direct contact. One axon is not directly touching the next dendrite, it's just really close. There is a gap there, and there's chemicals that are going to flow across that gap in order to signal off the next neuron. Just because an action potential goes down this nerve cell from here to there, and the action potential arrives at this cell, that doesn't mean that this cell is then going to take it and pass it on down to this cell. It may not. An action potential may or may not be propagated to the postsynaptic cell, depending on the amount of neurotransmitter released, the amount of acetylcholine, for example, that was released at this synapse here, and how is this cell here really ready to fire off again? Is it still in its refractory period? Is it still in its relative refractory period? We have a lot of neurotransmitters, and this is really an exciting field these days in medicine and in healthcare and everything, because these neurotransmitters are being discovered all the time now. We're understanding why certain things work in our brains and why certain things work in our bodies that we didn't understand before, because we didn't know what the neurotransmitters were. Couldn't figure it out. Research has helped us find a lot of them. Okay, so we have several classes of them. One of them is excitatory. Their whole deal is to activate the next neuron, cause excitation, cause depolarization of the postsynaptic membrane. Others are inhibitory. They may cause hyperpolarization of that postsynaptic membrane and therefore actually suppress action potentials. It would probably be nice sometimes to be able to take a pill that would suppress a lot of this incoming crap, right, that we have bombarding our brain all the time, just as a thought. So, the whole thing, these neurotransmitters are found everywhere in our body pretty much. There's more of them right there at the synaptic membrane, but it doesn't really matter about the neurotransmitter. It depends on whether or not there are the appropriate receptors there to receive that transmitter. Okay? It's all about the receptors. For example, acetylcholine usually promotes action potentials. It makes muscles contract, for example. It makes things happen. But in some places, like in the heart, acetylcholine slows your heart. It weakens your heart. It is an inhibitor in the heart. Why? Because the receptors that are located in the heart itself are when they are affected by the acetylcholine, they cause inhibition of the heart muscle contractions. So, we call this is the most common kind of synapse in our body. They're called cholinergic synapses, which means they release ACH, acetylcholine. Cholinergic synapses. And just so you know, keep this in mind, that all neuromuscular junctions with skeletal muscle fibers are based on acetylcholine. They're cholinergic receptors. Many synapses in the brain, from brain cell to brain cell, brain cell to brain cell, most of those are cholinergic as well and run on acetylcholine. All neuron-to-neuron synapses in the peripheral nervous system. In other words, once the message has left your spinal cord, it's no longer in the central nervous system, it's now out in the peripheral nervous system. All neuron-to-neuron synapses out here in the peripheral nervous system are cholinergic. And then finally, all neuromuscular and neuroglandular junctions of the autonomic nervous system. Remember the autonomic nervous system? I know I've mentioned it to you. That's the automatic nervous system that controls a lot of things that we don't ever have to think about. It has two divisions. Remember the sympathetic division, which is fight or flight, and the parasympathetic division, which is rest and digest. Well, the rest and digest receptors are all based on acetylcholine. What's the neuroreceptor in most of the sympathetic division? The fight or flight? What do we call that receptor? Adrenaline. That's right. It's adrenaline or epinephrine. Okay? And we would call those adrenergic receptors or adrenergic synapses as opposed to cholinergic. Okay. So, events that happen at a cholinergic synapse, well, the action potential arrives, it depolarizes the synaptic knob, calcium ions enter, that causes exocytosis of those little vesicles that have the ACH stored up in them. It throws the ACH out in, ACH goes across to the receptors, it causes depolarization of that postsynaptic membrane, and then eventually along comes acetylcholinesterase. Acetylcholinesterase breaks the acetylcholine back down into acetate, and choline reprocesses it, puts it back in vesicles so it can all happen again. Let's talk about synaptic delay. I think it only makes sense that if you're passing an action potential from this nerve here, passes down this axon to this nerve cell here, there's going to be some delay here at this junction, okay? And that is called the synaptic delay. And that is a very short amount of time here, but it's the amount of time that it takes for that exocytosis to take place, for those membranes to open up, to release that acetylcholine there, for it to go across the gap, for it to stimulate on the other side. Not very long, is it? Pretty darn quick. Half a millisecond. Half of a thousandth of a second. But, think about this. How fast would you like for a reflex to work when you step on a pin to get the message, pull your leg up off of it? You would like that to go pretty quick, right? Because every millisecond you stay on that pin is that much farther into your foot that pin goes, right? So, what that means is that we have some reflexes, and that one with the pin is an example, where it only has one synapse. In other words, the nerve, the sensory nerve that feels the pin in your foot sends a message up here to your spinal cord, that long, but it's a fast fiber. It synapses on one muscle neuron, and there's one synapse there, and then that muscle axon sends a message back down to your muscles in your leg that make your leg lift off. Okay? That's called a unisynapse, or just a single. It's a reflex that only involves one synapse. That's the fastest kind. Most of our synapses most of our reflexes are not single synapse like that. They're usually several, but they're still pretty fast. Synaptic fatigue is a term that means you've run out of neurotransmitter, you've run out of acetylcholine, you need some time to rebuild it all back up, and that synapse is probably going to have to be inactive until the acetylcholine is replenished and built back up. Now, we have now recently learned that there are many,
Talking about the slide before, isn't it possible for it to be oversaturated as well? I thought I read something about that.
that would be like tetany, if there's too many stimuli, and there would be too much acetylcholine. I'm not quite sure what you're asking.
Well, synaptic fatigue, like where you can't do like any more because like you've reached the full.
That would be like tetany. Yeah.
Sorry.
No problem. Okay, so we have these other things that we now know are neurotransmitters. We didn't know that there were other medicines besides adrenaline and acetylcholine. That's all we thought there were. But now we know there's about 50, we've identified 50. I bet by today it's up to 300 in the last few years since this slide was made. Some of them are amino acids, some of them are peptides, some of them are prostaglandins, some of them are ATP, and some of them are dissolved gases. Let's talk about this for a minute. So here's some other ones that are important other than acetylcholine. I already mentioned to you norepinephrine or epinephrine. Those are the adrenergic receptors. Dopamine is a drug that we use a lot in the intensive care unit. Ambulance guys, anybody here on an ambulance? You have dopamine on your ambulance? You still have epi? Still have epinephrine, yeah. Most of them are moving away from epinephrine and moving into dopamine because it's more specific for the heart usually. Another one is serotonin. Anybody heard of serotonin before? and watch TV at night if you haven't. I'll talk about serotonin in just a minute. Maybe I'll talk about it now. Serotonin is a drug that is used in your brain, and it sends signals from your brain, from one brain cell to another brain cell, that generally make you feel pretty good. Okay, so serotonin is a good thing. We could all get high on serotonin here today if we could shoot it right into our brain. Okay, and we'd feel really great. So, what what the researchers have found is if they can find a way to increase the amount of serotonin in people's brains, they'll feel better. Make sense? Well, they came up with a group of drugs that they call SSRIs. Anybody ever heard of those? SSRIs. SSRIs. Selective serotonin reuptake inhibitors. In other words, these drugs select serotonin and they prevent it being reuptaken out of the brain and put out into the regular circulation. They're inhibitors of the things that want to reuptake it. Okay, so if you inhibit the drugs that take up the serotonin, that means there's going to be more serotonin left in your brain. Okay, SSRIs. There's a whole huge class of them now. It includes things like Prozac and all the antidepressant drugs. Anyway, you know there's a bunch of them. They're in your book somewhere too. Personally, we used to sit around in the Dells and in the doctor's lounge and say, you know what, everybody in this town is so damn depressed. We need to put SSRIs in the water system, so everybody would get some, so everybody would feel better. And just so you know, I've taken SSRIs before, especially in the winter when the days are so short and there's not enough light. I think I suffer from light affective disorder. That's why you'll probably notice I usually have the windows open in the rooms. I like light. But I used to, every winter, I would just kind of start feeling blah and not all that great, and nothing really made me all that happy. And I went to my doc and he said, here, try this, and I would take it. I can't remember what it was called. It wasn't Prozac, but it was one just like it. And I would take it, and I wouldn't feel a single thing for about 10 days. And then I would be thinking about it 10 days later, and I would think, hey, you know, I feel a little better. I don't feel like I have this big weight on my shoulders anymore. Everything isn't bugging me to death. And I think it really did work. And I would take it until springtime usually, and then come springtime when the flowers came out and the sun came up earlier and all that stuff, I usually quit it every year. But I think these antidepressants are wonderful. And for people with severe depression, they really work. They really, really work. And if you've never seen it happen, and I know none of you probably have, but it's pretty miraculous. I used to use a lot of antidepressants in my surgical practice, especially in cancer patients. I would operate on them, and we'd say, well, we think we got it all. Things are looking good. And here's these pain medicines to take because I know it hurts because I did this big operation on you. And they come back a week later and they say, Doc, I'm just not happy. You told me you might have gotten it all, but you might not have. And all I do is worry about it all the time. I'm depressed. I can't sleep. I can't think. And I prescribe antidepressant. I give them a month's worth, come back at the end of the month and say, well, how are you feeling? You feel better? Yeah, I really do. I just don't feel like the weight of the world is on my shoulders anymore. And I had patients who kept taking the antidepressants for, one guy took them for 15 years.
Is that bad for your liver?
No, it's not.
Does that make you tired?
Does it make you tired? No, I think it gives you energy.
Do you think like relatively short-term use of pain medication can also like mess up the receptors?
Well, sure it does. Pain medicines are really tough, but when you do surgery, you've got to give pain medicine.
Oh, I understand that. I'm just
Yeah.
asking like.
Oh yeah, that's part of it.
It does screw up.
Oh sure. Yeah, pain medicines are bad deals. So anyway, norepinephrine, epinephrine are released by adrenergic synapses. Adrenaline, adrenergic, that's where that comes from. Dopamine is really big in cocaine use and Parkinson's disease. The people in Parkinson's don't have enough dopa, and so you've seen Michael J. Fox when he wasn't taking his medicine just right, he's kind of like this, and he has a hard time talking. Muhammad Ali is the worst case. Muhammad never takes his medicines right. That's why he's always kind of screwed up. So dopa makes you feel better. That's why people take cocaine, because cocaine is a tremendous releaser of dopamine. You take cocaine, it causes you have all this dopamine in you, and it causes it to be released, and you have this incredible high that feels wonderful. The problem is that you then spend the rest of your life trying to get that high again, because you never have that much dopa around again to release, and so people become cocaine addicts, trying harder and harder to get high again, but they don't really ever get as high as they did the first time. They get somewhat high, but there's always this striving to remember how good it was the first time. That's why crack cocaine is thought to be addictive for life the first time you ever use it. Crack cocaine supposedly gives you such an incredible high the first time you use it that you will then give up everything in your life trying to get that high again. And that's why it's so addictive. So don't try it. Serotonin I already talked about. GABA is another one that I don't teach you much about because I don't know much about it. Neuropeptides, opioids. Opioids are drugs that are derived from opium, which means morphine, which means cocaine, which means all opiate type drugs. They're wonderful for relieving pain. Unfortunately, they're also horrible in terms of creating addiction because people like to feel that way. 4 classes of them here. I don't care that you know this. I do think you should know what endorphins are. Endorphins are self-made orphins, morphine-like substances. And I used to run a lot, and if I would go out for a run, I would run about 8 miles, and I would feel like crap. But then if I ran another mile, I would just start feeling great. And I would get this second wind or second high, this endorphin high from endorphins. And then you can run another 6 or 8 miles, and you're just high as a kite and feel great. It's caused by release of endorphins into your own body. I'll leave it at that. There's a term that they talk about in the book a lot called information processing. Information processing is how do we think? How do we process all this information that comes into us? How do we decide what's important and what's not? Well, a single nerve cell, when it receives an action potential, deciding whether that action potential is strong enough to pass along or not, is the first level of information processing. That's the simplest form of information processing. I've talked about the fan and the projector. I've talked about the weight of your shirt on your shoulders. Those are things that our brain cells just kind of filter out because they don't seem that important. Okay, so stop the chapter at this point. How neurotransmitters and neuromodulators work. 419. I think that's maybe the old edition page. But I don't expect you to know much about these neurotransmitters, other than kind of what I've said here. Know about the morphines, know about the endorphins, know about dopamine, know about DOPA, because that's the drug seen in Parkinson's disease. The problem with Parkinson's disease, just so you know, is that people don't make enough DOPA. And so we give them DOPA. The pill they're taking is actually called L-DOPA, okay? The problem with it is that it's absorbed, it goes to a high level, and then it goes back down, and you've got to take another pill. And you got to take like 6 a day, you know, so you got to be watching your watch and taking them every 6 hours in order to maintain a level of DOPA that allows you to function. If you go a little long between doses, you get tight. If you take it too often, you get sleepy sometimes. So dosing has been the problem. The pharmacologists are working to try to come up with a form that would be slow release. The way we want to all take pills, I don't know how many of you take pills, but the best way to take pills is once a day. If you can take a pill once a day and you can pattern your life so that every morning when you get up and brush your teeth, you take your pill and that's it for the day, you're in pretty good shape. We know that if you have to give somebody a pill that they have to take twice a day, the chance of them actually taking it twice a day drops to about 50%. If they have to take it 3 times a day, it drops to about 30%. Actually take it 3 times a day on schedule. And if they have to take it 4 times a day, it drops down to about 10%. Only about 10% of people are controlled enough, whatever you want to say, to actually take their pill every 6 hours, if that's what it's supposed to be. It's just, we don't take pills well. So obviously the ideal is to take it once every day. Better yet would be once a week or once a month. And for some of the drugs, they've found ways to do that. The last thing that I wanted to mention on here that I didn't, you can pass those around. This slide right here, I said I didn't really talk about it. This dissolved gases, some dissolved gases work as neurotransmitters. Well, the best one of those is a gas called nitric oxide. Nitric oxide, I think it's NO3. And the nitric oxide is a gas that we found can be released by certain drugs that can be given. And nitric oxide only affects certain receptors in the body. And the receptors that they work on are in men, and they are the receptors that change the amount of blood flow going to the penis. So the class of drugs, which we now think of as Viagra and Cialis and all those erection drugs, work off of a gas, basically. The pill that you take releases the gas circulates through your body to those particular areas, because that's the only place in your body that has those certain receptors, and causes you to have a erection. I hear. It's the only drug in history that they could charge whatever they wanted to and sell as much as they possibly could. It's made billions and billions of dollars for the drug companies, because men will pay anything for an erection.
I was at Walgreens, and this guy that was in front of me was like cursing like a sailor because he was trying to get Viagra.
Yeah.
It was like $1,100.
Oh, it's crazy. Yeah, the prices are ridiculous.
He's like, can you cut my
$30 a pill, I've heard that. The newer drugs, Levitra and Cialis, they're coming down in price. Pretty soon they'll go into the generic. After 7 years, they're supposed to become generic, and then they'll drop way down. And by that time, the drug companies will have made their money back that they spent researching it. Okay. Are you ready?
I know, I got one.
All right, question number 1. Clear your desks off, please.